All right. Um, what's the function of the hard palate?
Separates the oral cavity from the nasal cavity.
Okay, separates oral cavity from the nasal cavity. Um, provides a surface against which the bolus of food can be manipulated. It can be flattened or scraped, something like that. Um, what about function for the soft palate?
It like helps close off something from the nasal pharynx with swallowing.
Okay, good, good. So like the soft palate and the uvula during swallowing helps to block the nasopharynx so that the food doesn't go into the nasal cavity. It goes into the the oral pharynx and then the laryngopharynx. Good. All right, what are these? Good, the palatine tonsils. Good. What do those do?
Immune surveillance of air and saliva.
Good, right? Immune surveillance of air and saliva. That's great. Good. What is this organ? The tongue. Good. What does the tongue do in the context of digestion?
It's the bolus for mechanical digestion.
Exactly, right? So aids in mechanical digestion of the food, manipulates the bolus within the oral cavity. Good. And then this,
Lingual frenulum.
good, the lingual frenulum. What does that do?
Could you just say like attaches tongue to the inferior floor?
Yeah, attaches the tongue to the inferior floor of the oral cavity, restricts the movement of the tongue within the oral cavity. Yes, good. All right, so that's oral cavity. This is also oral cavity. What is this again? Good, the lingual frenulum. Good. What about this one? You know what? I'm sorry. We don't learn about that gland. Ignore that. Yep, it's red. What about this thing? Not sublingual, but submandibular duct. Good. Yeah, this is the submandibular duct. There's like either one or two, and they're really large. The sublingual ducts, there's like multiple of them, and they're much thinner and smaller. Okay, what's the function of the submandibular duct?
Like extruding
Okay, to move like saliva from the gland into the oral cavity. The saliva has like a serous and a mucus portion. Yes, good. All right, which gland is this? Good, parotid gland. Parotid, that word is para, otid, and then get rid of the A, so it's parotid, and otid means ear, so it's next to the ear. Para is next to, so it's the parotid gland. What's the function of the parotid gland? Or how about what kinds of cells, glands, make up the parotid gland? Good, the serous acini, and it's mostly serous acini. Good. What is this? Good, the parotid duct. Good. Yeah, all the ducts are the same to transport saliva to the oral cavity. Which gland is this? Good, submandibular gland. Good. Yeah, here's the mandible here. So it's under the mandible, so it's called the submandibular gland. What's the ratio of serous to mucus acini? It's like about 50-50, exactly. Good. All right. Here, let's just name these again. What is this? Good, the soft palate. Here. Good, the uvula. What is this thing?
Pharyngeal tonsil.
Good, the pharyngeal tonsil. What's another name for it?
Adenoid.
Good, adenoid tonsil. Good. Yeah, the functions of all the tonsils is the same. To surveil the, well, I guess technically this one is not surveilling food, it's only surveilling air. These ones down here are surveilling food and air. All right. What is this one?
Palatine.
Good, palatine. Here. Good, lingual. And here's some landmarks. What is this? It looks like a bone. Good, the hyoid bone. Good. Yeah, that's an important landmark for like the separation between like this cavity or this space and this space. What is this space? Good, the oropharynx. And then this one? Good, the laryngopharynx. Good. And then what is this inferior to the laryngopharynx? The esophagus. Good. Let's not do the airway. Let's just leave that for next time. And yeah, actually, let's keep going then. Okay, which gland is this? Parotid. Here, what is this? Parotid duct. Which gland? Sublingual. Oh, it should be. And then these. Good, the sublingual ducts. What is this one? Good, the submandibular duct. Good. Yeah, the submandibular duct, it pierces the sublingual gland and then runs medial to it. And what is this gland? Good, the submandibular gland. Good. Okay. All right, let's go to so after the oral cavity is the oropharynx, and then the laryngopharynx, and then the esophagus. What's the organ after the esophagus? Good, the stomach. What is this region of the stomach? Good, the fundus. What about this region? Good, the body. And then what about this region? Good, pyloric. And oh, I don't know what this one says. Oh, this is in the lab manual too, right? The cardia. Okay. Yeah, the cardia is this region of the stomach that connects to the esophagus. So yeah, that's what that is. That's the esophagus. And what is this portion of the stomach? Good, the lesser curvature. This portion? Good, the greater curvature. What's the function of the lesser curvature? Good, connects the lesser omentum, connects the stomach to the liver through the lesser omentum. Good. What about function for greater curvature? Good, connects to the greater omentum, so that's what this is. Connects the stomach to the transverse colon through the greater omentum, that's another one. And the fundic, the fundus of the stomach and the body of the stomach is the same histologically. So when you're looking at those slides that say fundic stomach, you could really be looking at like this whole region here. there's only three regions of the stomach histologically: the like fundic slash body, and then the cardiac, and then the pyloric. All right. Yeah, what is this structure again? Good, the lesser omentum, good. All right. And then you guys can do the other ones later. Yeah, here's the stomach, just again. And let's see. What is this region? Good, the fundus. This region? The cardia. This region? The body. Oops. And then this region? Good, pyloric. I didn't block it out. Oops. What is this? Greater curvature here, lesser curvature. Good. All right. So that's the stomach. And then, or at least that's the stomach with the serosa still on it. And then if you take the serosa off, you can see, what is this layer? Like what is all of this layer? This third layer of the GI tract wall? Good, the muscularis, good. But the stomach has three regions within its muscularis. What is this one? It's not the oblique. Yeah, it's got to be the outer longitudinal layer. And then deep to that is this one. What is this one? Good, except it's called middle circular. And then this one? Good, the inner oblique. Good. So yeah, all of that is the muscular coat, the tunica muscularis, muscularis externa, yes. And then, what are these things? Good, the rugae. Good. What's the function of rugae? Good, to allow for expansion and like contraction of the stomach to accommodate chyme. Good. And then what is this? Good, the pyloric sphincter. Good. What's the function of any sphincter in the body? Or any sphincter of the GI tract?
Just control movement of contents.
Exactly, right? To regulate the movement of the bolus or chyme to the next part of the GI tract. Yes, good. Oh, that's the fundus. Okay. Okay, after the stomach is the small intestine. What's the first part of the small intestine? Good, the duodenum or the duodenum. Good. I don't know where that is, but let's just name these things. What is this again? Good, the pyloric sphincter. And these sphincters are a thickened region of which layer specifically? Good, the inner circular layer. Good. What are these? Good, the plica circularis, or circular folds. What is this? Good, bile duct or common bile duct. What is this? Which organ is this? Good, the pancreas. So these are ducts in the pancreas, so they're pancreatic ducts. But which pancreatic duct is this? It's called the accessory pancreatic duct. What about this one? It's called the main pancreatic duct. That actually shouldn't be green. I don't think that's in the lab manual. Don't look. What is this? Good, the major duodenal papilla. Good. All right, so then what's the function of either of the pancreatic ducts? Good, to secrete pancreatic juice, to carry pancreatic juice into the duodenum. And what kinds of things are in the pancreatic juice? Not bile, no.
Is it amylase?
Okay, amylase.
Yeah, and then,
Let's say,
something that multiple fats, right? Also, and then
yeah,
that's
that's the bile coming from gallbladder and liver. So, so maybe we can summarize it as like there are digestive enzymes and bicarbonate. Those are the two most important things, right? And then the digestive enzymes are for the like macromolecules in the food. And there's 4 macromolecules. What are the 4 macromolecules? Carbs, lipids, fats. Oh, sorry. No, because fats and lipids is the same thing. So carbs, lipids, proteins, and the 4th one is nucleic acids, right? Like DNA and RNA, like inside of cells. So the pancreas has enzymes to break down all of those things that we eat in the food. And so that's what pancreatic juice is, all those digestive enzymes plus bicarbonate. Why is there bicarbonate in pancreatic juice?
it's like a buffer with acid.
Exactly, right? Because the chyme coming from the stomach is very acidic and it should be neutralized. Otherwise, the enzymes, digestive enzymes coming from the pancreas won't work because they don't work in low pH conditions. Also, the enzymes along the brush border of the duodenum won't work if the pH of the chyme is too low. And so the pH of the chyme should be raised. It's raised by the bicarbonate coming from the pancreas. How else is it raised? Like which other modification in the duodenum raises the pH of the chyme? Think of the like submucosa, the duodenal submucosa. What's in the submucosa of the duodenum? Good, the Brunner's glands, exactly. What do the Brunner's glands do? Okay, but it's not just bicarbonate, like the bicarbonate is suspended in what solution? Mucus, right? So it's to secrete mucus that is rich in bicarbonate. Okay, let's do, so here's tunica muscularis, like both of these layers. What is this layer? Good, the outer longitudinal. And this one? Good, the inner circular layer. Good. What is this then? Yeah, it's got to be the submucosa. Good. Okay. And then after the duodenum is the jejunum and the ileum. We can't really differentiate between those two on gross anatomy other than by saying that, well, this must be ileum because the ileum connects to which part of the large intestine? The cecum, good. So it's like if it's closer, like here, it must be the ileum, but if it's more up here, it must be the jejunum, because we don't learn about how to actually differentiate. And then after the ileum is the large intestine. What are the 4 parts of the large intestine? That's the colon. So the 4 parts of the large intestine are the cecum, the colon, the rectum, and then the anal canal. So which part is this? Good, this is the cecum. And then what is this hanging off of the cecum? Good, what kind of appendix? Good, the vermiform appendix. Yeah, we have to say vermiform because, well, this word means worm-like. Appendix just means like something that's hanging off, right? So it's like this is some worm-like thing that's hanging off, but like then you have these things that are also hanging off of the large intestine. So which appendices or appendages are these? Good, epiploic or omental. So yeah, this word is like plural for appendix. So there's two types of appendices, right? The vermiform appendix and then the epiploic appendix. And there are multiple epiploic appendices, or you can call them epiploic appendages. Okay.
What do the epiploic eat? What do they do?
They're not well studied, but what they are is like visceral peritoneum with fat inside. So fat storage, that's at least one thing that it does. But yeah, I don't think they really know what these are for, like specifically. Okay, what's the function of the vermiform appendix? Okay, good, right? So like for immune surveillance, it houses like beneficial bacteria, so that basically the way I think about it is that these, like the cecum and the appendix is like a little like petri dish for bacteria, like good and bad bacteria. And the vermiform appendix has good bacteria in there. So if like the good bacteria in the rest of the GI tract is like decimated because of infection or like antibiotics or something like that, then the good bacteria in the vermiform appendix can come out and repopulate the large intestine. That's one function. Yeah, it's also receiving like the chyme from the ileum. So it's like that might be bad bacteria, but you need to receive that bad bacteria so that you can develop like antibodies and like memory cells against the bad bacteria. That's also why there's a lot of like which cells at the base of the intestinal glands in the ileum. So which cells are at the base of the intestinal gland in the ileum that help to fight bacteria? Good, the Paneth cells, exactly, right? Because if like the bacteria in here had the opportunity, they would just crawl into the ileum and just start colonizing the ileum, and that's not good. So the ileum has all of these Paneth cells inside to help kill the bacteria if they ever wanted to do something like that. Okay. What is this? Good, the ileocecal valve, good. I think it says orifice. What about this one? Yeah, no, we don't learn about that. Yes, this is the ileocecal valve, good. It's a sphincter. So what's the function of the ileocecal valve? Okay, good, right? So it prevents the bacteria from going back into the ileum. Also regulates the passage of chyme from ileum into cecum. Yes? Okay, which part of large intestine is this? Good, the ascending colon. Here? Transverse colon. Oh, it should be that one. Here? Descending colon. Here? Sigmoid colon. And what's after the sigmoid colon? The rectum, good. And then it's not on here, and I don't think in the lab manual either, but is the anal canal. Is rectum in the lab manual? Not for gross anatomy? Okay. Okay, don't worry about that then. All right, what are these? Good, the Tenia coli, good. And then singular is Tenia coli. What is this?
Greater omentum.
Good, the greater omentum, good. What are these? Good, Haustra. That's another Tenia coli. So is that one. There's 3 of them, like on each side of the large intestine. And yeah, what's this is just the same thing, yep. All right.
How do you work the function for Tenia coli?
Creates the haustra. Yeah, contracts to create haustra. Aids in peristalsis because it's just a thickened region of the outer longitudinal layer.
Wait, I have a question about that.
Mm-hmm.
So peristalsis is like all the way through, it's not just from the esophagus like to the stomach, like it could be in like the small intestine and the large intestine?
Mm-hmm.
Okay.
Yep. Yeah, all the way. Yep. I think segmentation can also occur in the large intestine, but it's not as important in the large intestine because most of the digestion and absorption has already occurred. Mm-hmm. Yeah.
Are the haustra like a functional unit of the? I don't know what their function is.
Yeah, their function is to. I don't recall actually. It's not a clear function. It's something like helps to like compact the feces, something like that. I don't recall actually.
There's a little
Does any?
bumps that-
Yeah, the little bumps. Yeah, each one of these sections. Yes. Yeah. Does anybody have a good function for haustra?
Not really.
Okay.
So you're just calling it like it's just a bunch of Tenia coli.
Okay. Yeah. I don't know. It's doing something that we're not sure of. Yeah. All right. All right. Let's do the histology now. Okay, which organ is this? Okay, good. Which tissue is this? At the submucosa. That's a layer too. Good. It's skeletal muscle tissue. Good. Because most of the tongue is skeletal muscle tissue. Innervated by which cranial nerve?
12.
12. Good. What's the name of the 12th cranial nerve? Good, the hypoglossal nerve. Good. Yeah, it's called hypoglossal because it goes under the tongue to innervate the skeletal muscle of the tongue. So that's why the skeletal muscle of the tongue is here, under the tongue. What is this layer, this general layer? This is not in the lab manual, I don't think technically, but like if this is the lumen, then what's the first general layer of any region of GI tract? Good, the mucosa, good. And then the mucosa has regions within it. So what is this region? Good, the epithelial tissue layer, good. This region? Good, the lamina propria, good. And then what is the specific ET like here? Good, keratinized stratified squamous epithelial tissue, good. Yeah, that ET is forming, or like is on top of which structure? Yeah, which structure is this? Not, it's a papilla. Which papilla? Good, a filiform papilla. Good. Okay, good. What's the function of a filiform papilla? Good, grip, right? To like increase the contact surface area between the tongue and the bolus of food. Yes, good. Yeah, that's why the epithelium is keratinized stratified squamous epithelial tissue, because it's experiencing a lot of friction. All right. Which organ? Still the tongue. Good. What is this? Good. Good. A taste bud. Good. Good. Which two cells make up taste buds? Good. Gustatory cells and supporting cells. Good. What is this structure? It can't be a filiform papilla because it has taste buds. Filiform papillae don't have taste buds. Oh, you said fungiform? Okay, good. It's a fungiform papilla because the taste buds are on the top, and only fungiform papillae have taste buds on the top. Good. What is this structure? Good. That's a filiform papilla. Good. What is this specific layer? Good, the lamina propria. And what is it made of? Good, areolar CT. Good. What is this tissue? Good, skeletal muscle tissue. Nice, good job. Which structure is this? Why is it a foliate papilla? Nice, good, good. Yeah, so like here's a papilla, and then there's taste buds on the sides. So it's not a filiform and it's not a fungiform. So it's either a foliate or a valate. But next to this papilla is another one that's identical to it. So the only papillae that do that are foliate papillae. Because if this were a valate papilla, then next to it you would see like that ring structure and then filiform papillae. You wouldn't see another of the same papilla if this were a valate papilla. So this must be a foliate papilla. Good. Okay, what kind of cell is this?
Mucus.
Good, a mucus cell. What kind of mucus cell? What if I told you this is inside the oral cavity? Good, intrinsic, right? So it's an intrinsic mucus cell. What about here?
Serous.
Good, intrinsic serous cell. Good. And like which structures are they forming? Yes, salivary glands, intrinsic salivary glands. But what's this specific like? What's the specific structure, form, shape of this salivary gland? It's called a secretory unit or an acinus. Let me show you a picture. So we're looking at like a cross section through this. So it's like, this is what the salivary glands are made of, whether they're intrinsic or extrinsic. It's a bunch of the secretory cells and then the duct. And then the duct has different parts to it. We don't have to know that, but we do have to know that this like ball portion down here, it's called an acinus or a secretory unit. And the acinus or secretory unit is made up of, well, this one is showing like both cell types, but we're only learning the ones made up of one cell type, right? So either all of these cells are serous cells or they're all mucus cells. So if these are all serous cells, then this would be a serous secretory unit or a serous acinus. If they're all mucus cells, then it would be mucus secretory unit or a mucus acinus. So this is the structure of all of the salivary glands, intrinsic or extrinsic. Yes. So yeah, going back. Because glands, exocrine glands and endocrine, are cells, a bunch of cells together, right? So it's like the individual component is the cell, and then all of them together is the secretory unit, or the acinus. All right. All right, which tissue is this? Good, skeletal muscle tissue, good. Yeah, we know it's skeletal muscle tissue, well, because, well, this is the tongue, and just looking at the cells, you can see that, like this one, there's like so the nucleus is like pushed to the side, like there's one. So this is skeletal muscle tissue. What kinds of cells are these?
Serous?
These are mucus cells, intrinsic mucus cells. What about these ones? Serous. These ones are mucus because the cytoplasm is like frothy, bubbly. These ones are serous because the cytoplasm, you can't really tell right now because the resolution is poor, but the cytoplasm has like granules in it, right? Little dots. And so don't rely on the stain color here. Let me show you a different one where, let's just go back to this one actually. So like in the previous one, the mucus cells stained darker, but here the mucus cells are staining lighter. So don't rely on the color, but rely on like the texture of the cytoplasm. Like, does it look like frothy, bubbly? If yes, then it's a mucus cell. If not, then it's a serous cell. Okay, and then let's just look at a different one. This is higher magnification. So here you can like really like see like the granules inside the cytoplasm. Here are like the white spaces. So what do these cells secrete? Or like, yeah, what do these cells secrete?
Serous fluid?
Right, it's like a serous fluid, yes. And then what's inside of this serous fluid?
Saliva?
It has to combine like with the mucus to make saliva, but what kind of enzymes? Hmm? Hmm? Lysozyme? Yeah, we're in the oral cavity, so how do we have to start digesting the food chemically, like with these cells. How do we, which macromolecule are we digesting? Good, carbs. And like which enzyme does that?
Is it
Amylase,
Okay.
good, right? Amylase. Like in starch, starch has like two glucose polymers. One of them is called amylose, that's why they named it amylase. The other one is called amylopectin. So amylase breaks down starch, polysaccharides, carbohydrates, all that is the same thing. So these cells are the ones secreting the enzymes. What are these cells secreting? Just mucus, right? What does mucus do? Okay, good. Exactly. So like it helps keep the like contents of the bolus together, all of the different food particles and the enzymes. It reduces the friction of the bolus against the epithelium of the GI tract or the chyme once it gets into the stomach and distal to that. Yes, good. Which structure is this? Why is it a valate?
It's a different size
how about, I think, so you're not wrong.
One is not like
it's
the
a-
others.
It just doesn't look like it. No, it's not giving foliate, no.
It's not kidding.
You can use the size, so you can say, okay, well, like, we see a lot of tissue, so this is like low magnification, and it's taking up like a lot of the field of view, so it must be a large papilla, so it's a valate papilla. Yes, you can use that. You can also use, well, there's taste buds on the sides, so maybe it's a foliate, but no, because if this were a foliate, you would see another one of these right here, and you don't. So it's not a foliate, so it must be a valate. It's not filiform because filiform papillae don't have taste buds. Okay, good.
Jordan,
Yeah.
are these the ones that you can find on your tongue?
Hmm.
Those are the ones that are really pronounced, correct?
Yes.
Okay.
Yep. Yeah. In the back, they form a V. Yep. Hmm. All right. Which glands are these? What kind of mucus? Right, intrinsic mucus glands. Good. Yeah, I think we have to say intrinsic versus extrinsic because, like, there are two sets of salivary glands, right? Like the ones inside the oral cavity and the ones outside the oral cavity. And their structure is the same, right? They both have these serous and mucus cells. But these ones are, since they're inside the oral cavity, intrinsic mucus cells, intrinsic mucus glands, versus the ones outside would be extrinsic mucus cells, extrinsic mucus glands. I don't think it says extrinsic, though, in the lab manual when it's describing the types of cells that make up the extrinsic salivary glands. Does it?
No, it just says secretory units.
Okay,
It just has it like under the bigger category of like extrinsic salivary gland.
got it,
It doesn't say
got it, got
So
it.
you have to know.
probably.
So like with the
But it
parotid
does-
gland,
Good, yeah,
we could say extrinsic serous glands.
yes.
What's the difference?
Extrinsic means like outside of the oral cavity in this case, and then intrinsic means inside of the oral cavity. Yeah, I think we should say intrinsic mucus or intrinsic serous for these, and then for the other ones, well, the lab manual just says serous secretory units and mucus secretory units. Okay. Okay, which glands are these? Good, the serous glands of von Ebner. Good. So these are just the intrinsic serous glands associated with valate papillae. If you can't see the valate papilla, then we don't know. These could be any intrinsic serous glands, because you have these intrinsic salivary glands in the tongue, in the soft palate, in the mucosa of the cheek. So we don't know unless we can see the valate papilla. Yeah, we don't know if these are serous glands of von Ebner unless we can see the valate papilla. All right, what kind of tissue is this? Good, skeletal muscle tissue. Good. All right. All right, which structure is this?
It'd be a fungiform
Why is it a fungiform?
because it looks circular on the top and there's little other ones next to it. Or maybe am I in a different, oh no, I'm in a different organ, I don't know.
What are these?
Oh, those are taste buds.
Those are taste buds, but they're on the sides.
Yeah.
So can it be a fungiform or a filiform? Nope.
No.
So it's not fungiform, it's not filiform. It's either foliate or valate. If it's foliate, which should you see like right here?
I see another one.
Another one. Is this that?
No.
No.
It's a valid.
So it's a valate.
Wow.
Yep.
Okay.
It's a different, it's
Yep.
Very nicer looking. All right.
Yeah, don't use shape or like I think you should use the taste buds. Like where are the taste buds? If there are none, it's not filiform. If they're on the top, it's fungiform. If they're on the sides, the second question you ask, what's next to this papilla? Another one? If no, then it's a valate. If yes, then it's a foliate. And that's probably like the fastest and easiest way to differentiate between the papillae. Yeah. Yeah, because then what if the question was like, ID these glands? What kind of papilla is this?
Oh, it's a valate.
Right, so what kind of glands are these? Right, serous glands of von Ebner. Yep. Mm-hmm. All right. Okay, what kind of cells are these? Or which organ is this? Why is it the pancreas?
Because there's no lumen and it's not the salivary glands.
Okay, how do we know it's not a salivary gland? How about if this were the pancreas, which other, like what else should you see? Right, the islets. Where are the islets? What do the islets look like? They're lighter. Is there anything on here that looks especially lighter? Like other than like the actual like white spaces and cracks?
I was thinking by 32, but like 32 looks more like a gland, so that's where that's what
This looks pretty homogeneous to me.
That's the parotid.
It's got to be the parotid gland.
Yeah.
Yeah, the parotid gland and the pancreas look almost identical.
Yes.
So here, let me show you.
They're false.
Exactly.
Don't let number 32 steer you wrong. See?
Yeah, which one is this?
See it.
Same thing.
Would it be the pancreas?
It's got to be the pancreas, yes.
There's your linger on.
Exactly. It's like, it's not homogeneous. Like, here, let's, like here, like that for sure looks different, right? Like this one for sure looks different than like these cells over here. Like it's obviously lighter versus
Oh, he has a linkers to it.
like, I don't know. I don't see like some obvious lighter clump of cells. Everything kind of looks the same. So that's how you differentiate between parotid gland and pancreas. You have to really look for those islets. That's probably the hardest like differentiation, right? The parotid gland versus the pancreas. All right, let's go back up. All right, so what kind of cells are these? Good, serous cells, forming which structures?
So for in the parotid, extrinsic salivary, or extrinsic serous glands.
Yes, but what's, like, what is the shape of these extrinsic serous glands? Like, which structures do they form? They're called secretory units, or acini, right? Yeah,
Yes.
that's what it says in the lab manual. So let's call them serous secretory units. I guess it really should say like extrinsic serous secretory units, because there are also intrinsic serous secretory units, or you can call them acini. Yeah. Okay, and then what is this? A duct. Nice, good job. All right, let's go.
So
Okay, which organ is this?
Submandibular.
Why is it submandibular?
There's a good ratio of mucus acini and the serous acini,
Um,
maybe?
I think it's
Sublingual.
why is it sublingual?
Because I think it's serous cells, but I don't think it's forming serous in it. Like, there's kind of like scattered serous cells, but
Yeah,
Yeah.
yeah, most of the cells are mucus cells,
yeah.
right? So it's like here and here. Yeah, it's like most of what we see in the field of view are mucus cells, and there aren't very many serous cells. So there's more mucus than serous, so it's sublingual. Yes, good. All right, here.
This one's submandibular.
This one is submandibular. Good. Why is it submandibular?
This one has both serous and mucus in it.
Exactly. Nice. Good. Which organ? Why is it the esophagus? Nice, good. Yeah, so this layer is non-keratinized stratified squamous ET, and this is not the oral cavity because, well, there's no papillae, and like this is not skeletal muscle tissue. We don't see the features like of the tongue that we learned about, so it must be the esophagus. All right, what is this major layer? Good, the mucosa. What is this specific layer? Good, the lamina propria. What is it made of? Good, areolar CT. What is this specific layer? Good, the muscularis mucosae. And what is it made of?
Does this one transition like the muscularis or no?
That's for the muscularis externa.
But not for this one. This one doesn't transition at all.
Yeah, no.
Okay. Smooth muscle tissue.
Good, smooth muscle tissue. Nice. Good, good. All right, let's do another one. All right, which, yeah,
Does it show the adventitia in that slide?
it
For the
didn't.
esophagus? Okay.
Yeah, that was only showing the mucosa.
Okay. You don't
Mhm.
think they would do a slide with adventitia? for
They
the
could.
esophagus?
They could. Yep. Which organ is this? And how do you know?
Hmm?
Oh, okay, good. The small intestine because of the villi. Good. And then which region of the small intestine?
So jejunum?
Good, the jejunum. Good. Yeah, there's nothing in the submucosa, right? There's no glands, there's no Peyer's patches, so this is the jejunum. Good.
It's just functioning.
Which organ? Okay, what about, so it looks like, okay, which layer is this? The mucosa here? This is the submucosa, because if this is the mucosa, then the ET layer and the lamina propria and the muscularis mucosa are all in the mucosa. So this is the submucosa. There's some giant undulation of the submucosa. So which regions of the GI tract have giant folds in the submucosa? Okay, the large intestine, yes, but we don't learn about like the names of those folds of submucosa. So like we can't use that. What about which other regions of GI tract have folds in the submucosa and we learned the names of them? Okay, the small intestine. What are they called in the small intestine? Good, plica circularis. What about another region?
Stomach.
The stomach. What are they called in the stomach? Good, rugi. So it's like, okay, this is stomach or small intestine. What's the difference between stomach and small intestine? Good, villi or no villi. Small intestine has villi, stomach doesn't have villi. Do you see villi?
Does the stomach not have goblet cells also?
Stomach doesn't have goblet cells. Yes, that's correct. But we're kind of far to differentiate like between cell types. This is like the lowest magnification possible. What about like here and like here? Like, are these villi? No. So is this the small intestine? No. So this is the stomach, right? So that's one way to do it. Is we'll look at the submucosa and know that like the submucosa has folds, has like undulations in the small intestine and the stomach. In the small intestine, they're called the circular folds or the plicae circularis. In the stomach, they're called what? Rugi, right? So this is like one ruga, a singular ruga. And yeah, the epithelium, like there are no villi. Like the villi would look like, they would almost look like these over here, but they would be more pronounced. They would like really like be up there, you know? Like very obvious, like finger-looking structures, right? But like over here, like they're just not, yeah, these aren't villi here, because let me show you villi. They're not giving villi.
That would be simple columnar.
Yeah, these are villi, right? Like, look how much it extends up, and then it goes down, and then like another one, like these very tall column-looking things, right? So it's like over here, like you can see like the white space in between, and like this whole thing, right? Like that's a villus, right? Versus like over here, like maybe. But then, like, what happens, like, over here, you know, where it's like that's completely flat, and then that's just like a tiny, it needs to be like that, right? That's kind of what we just drew. So these aren't villi. So no villi, but fold and submucosa equals stomach from, like, low magnification. Okay. All right, let's do another one. Which cell is this? Good, a parietal cell. What does it do? Good, it secretes hydrochloric acid and intrinsic factor. What does hydrochloric acid do? Okay, lowers the pH, good. And that denatures proteins, yes. What else does it do? Good, it activates pepsinogen. What else does it do? Kills bacteria, nice, good job. Which cell is this? Any of these. A chief cell, good. What do they do? They secrete something. What do they secrete?
Carcinogen.
Good, they secrete pepsinogen. And what else? That's the parietal cell. Good, lipase, gastric lipase, good. What does lipase do? Gastric lipase do? Good, chemical digestion of lipids. Let's go back to parietal cell. What does intrinsic factor do?
Binds to B12 to be absorbed.
Exactly. And then where is it absorbed? I don't know if we even have to know this. In the small intestine. Good, good, yes. In the ileum, in the very distal part of the ileum. Good. All right. Which organ? Good, the liver. Which tube is this? Not the bile duct, but the bile ducty wall, right? Because the bile duct was that tube we saw on gross anatomy, but it's coming from smaller bile ducts, so they're called bile ductules, or branches of the bile duct. What's the function of the bile ductules?
Transport bile from hepatocytes to bile duct.
Nice.
to water.
Nice, good, good. To transport bile from hepatocytes to the bile duct to the duodenum. Good. And what does bile do? Good, it emulsifies fats. Good. Is that mechanical or chemical digestion? It's mechanical. It's mechanical because what is chemical digestion?
Like actually breaking bonds.
Exactly, right? Like the actual hydrolysis of the bonds between molecules, right? Like the actual cleavage of chemical bonds versus mechanical digestion is like a physical separation, right? It's like, somebody explained, it's like you have a bag of cotton balls and you pull out all the cotton balls and then you're separating the cotton balls into they're individual cotton ball, but then the actual tearing the cotton ball is chemical digestion. But just pulling out the cotton balls and separating them from each other, that's mechanical digestion. And so bile only does the taking the cotton balls out and separating them from each other, and then lipases actually rip and tear up the cotton balls. All right. Which tube is this? Good, branch of the hepatic artery. Good. How do you know? It has simple squamous ET, but there's another, this tube has simple squamous ET. So does this one. Okay, the lumen is smaller. The lumen isn't as collapsed as this one. So this one is more like a circle. This one is more like an oval. So it's the circle collapsed. The tunica media is thicker. So this pink here, you can see some pink, but here, it's really hard to tell, but it's here's the endothelial cell, and then where's the pink in between tunica interna and tunica externa? There really isn't much. So this must be a venule, this must be an arterial, or this is a branch of hepatic artery. This is branch of what? Right, branch of hepatic portal vein, right? Why is it not branch of hepatic vein?
Hepatic vein like drains the liver.
Exact-
It's symbolic.
Exactly, right? The portal vein supplies the liver with blood, but the hepatic veins drain the liver of blood. Good. And what kinds of nutrients, or how about, like, what kind of blood is in here? Like, think of the nutrients and then the other things that are inside the branches of the portal veins. Sorry, say it again.
Deoxygenated blood.
Okay, so the blood is deoxygenated, yes. What else is in this blood?
Nutrients, proteins, and carbs.
Exactly, right. Proteins and carbs, because the portal vein is draining the GI tract, the stomach, all the way to the large intestine. So there's proteins, carbs. Are there lipids in here?
No,
Why?
because lipids they pass through there. They go through the lymph system.
Exactly. There's no lipids in here because the lipids got absorbed into the lacteal in the villus, and then the lacteal is a lymph vessel, and then all of that has to go through like cisterna chyli, thoracic duct, into the left subclavian vein, and then into the heart, and then into the arteries, and then it can get to the liver. So the lipids are in here technically, right? Not in here. And what else is in this blood, in the branch of the portal vein?
Like red blood cells, like hemoglobin waiting to be recycled.
Exactly, right? So because the portal vein is draining the splenic vein, splenic vein is coming from the spleen. The spleen is the red blood cell graveyard, so that's where red blood cells go to die or after they've been lysed. And then all of that blood, all of those red blood cell fragments and the hemoglobin is being sent to the liver so that the iron can be recycled, so that the heme can be recycled, so that the globin, the protein, can be broken down into amino acids and be put into new proteins, new plasma protein, or those amino acids like sent like back to the red bone marrow for hematopoiesis, like erythropoiesis. Okay, good. What else is in this blood? Yeah, in the branch of the portal vein. There's a lot of stuff. Think of like the GI tract blood has nutrients, but like what else might have been in the food that you ate?
Like toxins.
Good, toxins.
And that's possible to be in there, so.
Yeah, toxins or just drinking alcohol, right? That's a toxin technically. That's getting here. That has to be broken down into
Are
a.
vitamins getting there, or is that more of absorption in the small intestine?
I think it depends. Yeah, vitamins, yes, as long as they're water soluble.
Yes.
Yeah, it can't be fat soluble because then they would have to go through the lacteal. And pathogens too might be in here, right, in the food that you ate. And so which cells in the liver deal with the pathogens? Good, the Kupffer cells, exactly. Good, good. Yeah, so that's the function of this tube, yes? Yeah, the liver does a lot of things. Yep. Okay.
I'll put that on half the line.
It'll probably say like give like one function. Yeah.
No, I'm just kidding.
Yeah.
The, so if you scan out like you already did,
Mhm.
what are the darker lines? Could you say those are Kupfers, or are those something else?
This is connective tissue. These
Oh,
are,
like further out.
oh, like here.
Yeah.
Yeah, all this is also connective tissue.
Okay.
No, we can't see the Kupffer cells unless like you feed some like
Yeah,
toxin.
that's what it is, yeah.
Mm-hmm.
Okay.
Yep. Yeah, so they're inside of which spaces?
Sinusoid?
Good. The sinusoids. Nice. Good. Yeah, so these are these are macrophages, so they're like phagocytosis of pathogens. These ones also help with like hemoglobin red blood cell recycling too. All right.
Are they really like that color in a living body? Dark.
No, these ones are dark because I don't remember how it works, but they gave the liver, or like the mammal, something to eat, and then but there was like a lot of carbon in it, and so when it got to the liver, the Kupffer cells like ingested that, and that made them stain this color. Mm. Okay, which organ?
Large intestine?
Why is it the large intestine?
Because there's so many goblet cells.
Okay, good. Yeah, that's a great characteristic. So if the gland is like all goblet cells, it must be the large intestine. Also, it looks like there should be a villus here and like here, but it looks like it got like chopped off. So like the mucosa looks very like linear. It looks like very flat. So this is what the large intestine looks like. I'm not sure what all of this is. So ID this specific layer. That's like the general layer, the major layer. What about like this specific layer? Good, the ET layer of the mucosa. And then which specific tissue is it? Exactly. Good. Simple columnar ET with goblet cells and microvilli. Good. Yeah, what's the function of this ET? So now think of like, what's the function of the large intestine? Good, right. So water absorption is the main function. And to absorb water, it's useful to first absorb salt. So it's salt and water absorption. And what else is absorbed in the large intestine? Think of like, okay, there's a lot of bacteria in the large intestine. Like, what are those bacteria doing? Like, why do we need bacteria in our large intestine? Okay, good, right? So there's some like carbohydrate sources that we can't break down from plants because we don't have the enzymes. But the bacteria in our gut do have the enzymes. So they can break down those fibers into like vitamins, into like fatty acids. So that's also what the large intestine is doing, is absorbing like the metabolic products from the bacteria living in the large intestine. Yeah, that's why there are microvilli. Okay, and then what is this specific layer? Good, the lamina propria. What's it made of? Good, areolar CT. And what's the function of areolar CT?
Metabolic support.
Good, good. Metabolic support for the ET. And what else?
It's like
Think of, we,
yeah, it has like cells where there's like bacteria and stuff that can like create an immune response.
exactly, right? So for immune surveillance, right? Because we just talked about how there's all these bacteria living in the large intestine, and if given the opportunity, they would just colonize our tissues. So deep to the ET is a layer of areolar connective tissue that surveils the bacteria, and if they try to invade, an immune response can be mounted. So that's true everywhere, right? Deep to ET, there's a layer of areolar CT, but it's like especially important in the large intestine because there are so many bacteria living in the lumen. And then, what is this structure? Good. What kind of gland? Good. An intestinal gland or intestinal crypt. Good. All right. What is this layer? Good. The muscularis mucosae. What is it made of? Good. Smooth muscle tissue. Good. What's the function of the muscularis mucosae?
It's to manipulate,
Yeah. Why is it in the mucosa?
isn't it to like manipulate the mucosa so it can like create more surface area and, yeah.
Exactly, right? So it's like to move the mucosa so that like different parts of the mucosa can be exposed to like whatever contents are in the lumen, right? And so that's important everywhere in the GI tract. In the large intestine for water absorption, the vitamin absorption, the fatty acid absorption. In the small intestine for like all of those macromolecules being absorbed, the proteins and the carbs and the fats and the nucleic acids. Yeah, so that's the function, right? To move the mucosa so that some parts of the mucosa that like weren't in contact with the contents inside the lumen can come into contact with the contents in the lumen. Okay.
Jordan, are you recording this section?
Mm,
Okay, I have a doctor's appointment, I'm sorry.
okay, no, you're okay.
Okay, see you guys
All right, which organ is this? Good, the pancreas. How do you know?
Yeah, it's like the Langerhans.
Nice, the islets of Langerhans. Good. Yeah, so that's what this is. And what kinds of cells are these? Or which structures are they forming?
Serious assessment.
Good, serous acini, serous secretory units. Good. What's the function of these cells?
They produce that pancreatic juice.
Okay, good. Technically, they only produce like which portion of the pancreatic juice? Like the bicarbonate portion or the digestive enzyme portion? It's the digestive enzyme portion. Yeah, because yeah, the bicarb doesn't come from these cells. It comes from the cells in the ducts, like coming from the acinus. Okay, and then what's the function of the pancreatic islet?
Produce glucose and, no, not glucose, insulin and glucagon.
Good. So to produce insulin and glucagon. What does insulin do? Good. It lowers your blood sugar. Good. And like when the sugar is leaving the blood, where is it going?
Wait, sugar's leaving the blood, it's going to the cells.
Exactly. Good. It's going into the cells. Yeah, we're not like peeing it out or like it's not going into your sweat glands or something like that. No, it's going into your cells for metabolism. And what about glucagon? What's the function of glucagon? Good, right? The opposite. So to increase the blood sugar. And how is the blood sugar increasing? Like, where's the sugar coming from? Good, glycogen. And which glycogen? Because we have glycogen in the skeletal muscles and we have glycogen in the liver. Sorry, say it again?
Like liver stores, which one liver stores, isn't it?
Right, exactly, from the liver glycogen. Good. Yeah, not the muscle glycogen, because the muscle glycogen is for the muscles, so that the muscles can be like ready to act, move, metabolize when they need to. Okay, good. It also, never mind, that's fine. That's too much physiology. Okay, good. I hope so. Okay, only a couple more. We've done almost all the organs. Let's do this one. Which organ is this?
Small intestine.
Okay, small intestine, good. Which region of the small intestine? Why is it the ileum?
Because of the tires.
Exactly, good. Yeah, so here's the mucosa where, like here's the muscularis mucosa, which means that this must be which layer? The submucosa, good. And all of this purple is a lymphatic nodule, a giant lymphatic nodule. So this is a Peyer's patch, good. What's the function of Peyer's patches?
Immune surveillance.
Exactly, immune surveillance, good. Yeah, why are there so many lymphatic nodules in the ileum? And like not in the jejunum or the duodenum?
Because it's right before the cecum,
Ex-
like all the bacteria
ex-
is stored.
Exactly, right? So the bacteria in the cecum and the appendix might want to crawl back into the ileum. So if they do, then they encounter the Peyer's patches, and also like which cells at the bottom of the crypts?
And it's
The Paneth cells, exactly. Good job. All right. And which organ?
small intestine.
Small intestine. Which region?
Duodenum.
Why is it the duodenum?
Because of the Brunner's disease.
Good, right? So if this is the mucosa, then this is the submucosa, and there are all of these glands in the submucosa. So these are duodenal glands. Yeah, we know that this is the small intestine because these are villi. Only small intestine has villi. And let's name some things on here that we didn't talk about yet. What is this?
Myenteric plexus.
Good, the myenteric plexus. Yeah, you always find it in between which two layers?
The two muscular layers and the muscle layers, so the longitudinal and circular.
Good, so here's the outer longitudinal, and then here's the inner circular. Good. And sorry, let's talk about what's the function of the duodenal glands.
Secrete bicarbonate-rich mucus.
Good. And why is this mucus rich in bicarbonate?
To be a buffer against the chyme that's coming in,
Ex-
a lot of acid.
Exactly. And why is it important to neutralize the acidic chyme coming from the stomach?
To not like break down tissues in the small intestine.
Okay, so to protect the epithelium of the small intestine, yes.
Also like enzymes and stuff in there too.
Exactly, right? To prevent the denaturation of the enzymes coming from the pancreas, yes? And also the enzymes along the brush border of the small intestine. Yes, good. Yeah, here, this will be the last one. I didn't label it, but like here and here, what is this? So these are the duodenal glands. The pink is like collagen fiber. These are like nuclei of fibroblasts. But like, what is this other thing in the submucosa?
Like, mole?
Uh,
Or
not,
is it supposed to be like the mesenteric?
not the mesenteric plexus, but the. It's
Oh, submucosal plexus.
good. The submucosal plexus or the Meissner's plexus. Good. All right. What's the function of the submucosal plexus?
Innervate the mucosa.
Good. And
The submucosa.
and the submucosa. Good. Yeah. So all of this is innervated by the submucosal plexus. And then what's innervating all of this?
The mesenteric.
Good. The myenteric plexus. Good. Yeah. So the submucosal plexus has to innervate like the autonomic effectors, right? So like the glands and the smooth muscle. So the smooth muscle of the muscularis mucosae, the duodenal glands, the intestinal glands. Yes? Which cranial nerve is projecting onto these ganglia?
Vagus.
Good. The vagus nerve. Good. Which number? 10. Nice. Good. Yeah. So that's how all that goes together, right? That long preganglionic axon is coming down, and then it synapses on the postganglionic cell body, and all that is parasympathetic. And then the postganglionic axon goes out and innervates like the effectors, the smooth muscle and the glands. Yes. Nice.
So the submucosal plexus could be like anywhere in the submucosa, like it's not usually like, okay.
Yeah. I think usually it is like towards the base.
Okay.
At least like that's where the neurons are.
Mm-hmm.
But you can see it like in the middle.
Yeah.
You can see axons sometimes like coursing through. Yep.
Okay.
All right. That